Hello, and welcome to our lecture on eukaryotes. I will try and make this series a bit brief. We kind of already have gone over helminths, fungi, algae, and that in the intro lecture. But this is going to kind of send them home as we kind of proceed down metabolism in the coming weeks. So, we want to be able to compare and contrast prokaryotes and eukaryotes that are known to cause disease in various species with specific details from model species that we want to know. And we will classify organisms as prokaryotes or eukaryotes according to their cell structures, appearance, and taxonomy. Keep an eye out for those. Eukaryotic microorganisms include our protozoa, algae, slime molds, water molds, and we've already looked at protozoa and algae underneath our microscope under the pond water. We were able to see all the green algae and the protozoa. Alright, so when we talk about eukaryotes in the context of microbiology, we're mostly talking about human pathogens and vital human organisms. Vital human organisms are like fungi and things that break down things for us that we take advantage of. So without decomposition, we would be in, rather bad sorts. So we depend on some things for decomposition. That's what that vital human organisms mean, and that is what we study in micro. So we touched on this again. I just want to briefly go through it. Eukaryotic reproduction is usually through mitosis. This is mostly an A and P thing, but briefly, the cell will divide by first replicating its DNA. Once it is able to replicate a full set of DNA, it starts to split into two, and in order to do that, it is going to use the centrioles to pull that DNA into opposite poles. We label each of the phases, and by the time there are two full nucleuses, those cells will pinch, and we will have two daughter cells. So that is how most eukaryotic cells reproduce. We do have some exceptions that we note, some notable exceptions that we see coming up. So to finish this video, we'll talk about protozoa and why we learn about them in microbiology. Okay, and then here we have a dinoflagellate, a typical protozoa. And I love how they do these images. They make the image a TEM or an SEM on the left or the right, and then they had an artist drawing to kind of fill in the gaps and what it would look like there. So it's kind of like visually appealing, but also actual and factual. This is a typical paramecium, and you will note the cilia all along here, the little hairs along its body. They move to let it have motility that we discussed in our motility lab. They are eukaryotic, unicellular, and they do not have a cell wall. As you can tell, they have a unique shape, but no cell wall keeps that shape. It's just a cell membrane, and on the outside of the cell membrane are cilia. Okay, most protozoa either have cilia, flagella, or some kind of pseudopod, and they're all sorts of morphologies. A pseudopod is like an amoeba, and the amoeba moves by disassembling and reassembling its cell structure, cell skeleton, cytoskeleton, as it moves, and that is a pseudopod. Protozoa require moist environments. That's why pond water is a great place for them. That's why we filter our water. That's why we don't drink fresh water. We boil it first or filter it first or treat it first. Most live in ponds, streams, lakes, oceans. Soils also contain them, and obviously not a lot of them are pathogens. One thing to note, though, is that the trophozoite stage is the most infectious stage of a protozoa. So if you're going to get sick from one of these, you most likely caught it in the trophozoite stage. Okay, and then we also see that the cyst stage is actually known as the resting stage. And this guy, Trypanosoma, is what we'll see in the lab, actually. We actually see that this is African sleeping sickness, and they are caused by these little guys in our blood. And here we have a sexually transmitted disease up there, and those are both caused by protozoa. This is a rather cool picture of a protozoa eating another protozoa. This is a predatory ciliate devouring another ciliate, paramecium. So we saw the paramecium from a 2D image, and we saw all the organelles inside, and we could just barely make out the cilia, but here we can actually see the cilia, and we can see how these two ciliated protozoa are interacting. Okay, so they mostly absorb nutrients from other creatures is the main thing, except for dinoflagellates, which are photoautotrophic, which means that they get their energy from light, and they make it themselves. Finally, we have that amoeba that I was saying, that amoeba that disassembles its skeleton as it moves. We can have several types of amoebas, Naegleria being one of our most popular ones. And we have slime molds also that are classified as amoebozoa now. And just interesting to note that these are our pathogens, and that is our topic on protozoa. We've seen these in pond water. These are eukaryotic cells. They have eukaryotic structures. They have mitochondria. They have all sorts of things. And if you put a bacteria next to them, it would only be about yay big. Okay, so, or even smaller. So keep that in mind when we talk about eukaryotes versus prokaryotes, that there's a whole size difference too that we're dealing with. Our next lecture will be on fungi and other types of eukaryotes.